What was the ancestral state of the vertebrate urinary system? How did ancestral vertebrates excrete their nitrogenous wastes? In the most primitive type of kidney which exists today, the pronephros, there are actually different mechanisms through which nitrogenous waste can be excreted. First, nephric tubules which lack glomeruli, so they are aglomerular. They can filter the coelomic fluid as they open up into the coelom or even the pericardial cavity. And ciliated cells can thus transport coelomic fluid through the nephric tubule, and this can be excreted into the archienephric duct and released from the body. The nephric tubules can reabsorb water and salts and excrete additional waste and process the fluid additionally. Capillaries can exist, which are not in a glomerular capsule. They are external glomerular capillaries. They can release filtrate fluid, and thus nitrogenous waste from the blood can be released through these nephric tubules. Third, it is possible that nitrogenous waste can both pass from the coelomic fluid into nephric tubules through ciliated cells, but then also these same nephric tubules may be associated with glomerular capillaries so that they may receive nitrogenous waste from two separate sources. And then finally, the situation which not only exists in some pronephric nephrons, but also the nephrons of mammals, is that a glomerular capsule collects capillary filtrate without a connection to the coelom. The pronephros, which may contain all of these types of mechanisms to excrete waste, varies in how it develops in larvae and adults. In hagfish, not only is the embryonic and larval pronephros functional, but also the adult pronephros is functional. The anterior portions have aglomerular nephrons that are draining coelomic fluid, while the posterior nephrons lack a connection to the coelom, and the glomerular capillary is enclosed in a glomerular capsule. In lampreys, the larval pronephros is functional and is the only mechanism for excretion in young larvae. In most jawed fishes, the larval pronephros is functional, and in a few teleost species, the pronephros in adults is also functional. In amphibians, the larval pronephros may be functional, and a few nephrons from the pronephros may actually contribute to the adult kidney, perhaps one or two in many amphibians, but as many as a dozen in some caecilians. In reptiles, the embryonic and adult pronephros is not functional, and in mammals, the embryonic and adult pronephros is not functional and will deteriorate during embryonic development.